going to tell you about a project we've been working on for a few years on how bacteria survive in fluctuating conditions. And we've been using a microfluidic device we designed to study rapid fluctuating environments or slow environments and look at cells proliferating over long periods of time. A fluid flow brings in nutrients and also takes away cells. And from these kinds of experiments, we found you can recapitulate classic experiments of Monod in which you change the environment. For example, in a glucose to lactose switch, you see the lag phase, which lasts about an hour before cells are able to recover. But we noticed a few years ago that the lag phase disappears in fluctuating conditions. What this means is when cells first see lactose, they go through the classic lag phase, but after they go through another period of 4 hours in glucose, where they divide once an hour, the great-grandmother cell can actually remember that it experienced lactose in the past and avoids lag phases thereafter. The molecular mechanism that we've been studying is tied to the inheritance of stable lac proteins that are inherited from mother to daughter cells and then provide memory for a number of generations. So we've been investigating the molecular mechanism by targeting LAC-Z to proteolysis by attaching an SSRA degradation tag to its C-terminus. And what we discovered is that functional LAC-Z actually cannot be degraded by E. coli's native proteolytic machinery. So the mechanism we've been studying here is that the polypeptide folds to produce monomers. The monomers reversibly make dimers. The dimers then irreversibly build tetramers, and those tetramers are protected from proteolysis. So to probe that mechanism, we've been using the classic alpha complementation system to disrupt the tetramer and then recover its assembly. And in the meantime, we also found during these experiments that the unassembled LAC-Z monomers and dimers can actually aggregate in vivo and cause proteotoxic stress. So this classic system of LAC in bacteria in E. coli, which we can use to study memory, is also a nice in vivo model system to study protein assembly, aggregation, and degradation, and could aid in our understanding of protein misfolding diseases. And our lab works broadly on quantitative microbiology, evolutionary dynamics, and biophysical theory. And so I'd be excited to speak with people about many topics. Other projects we're looking at are stochastic switches in microbes as survival strategies, recombination in bacteria and viruses, and things related to biophysics of evolving populations. We have postdoc positions available as well. Contact me if you're interested in that, or you know people who are. And I'd like to acknowledge Dan Pollock, who's a PhD student in my lab who did this work, together with Takashi Nozoe, who's a postdoc. Thanks very much.